Hi there, I hope you are doing fine. This is Mr. Philip Kauma, the excellent biology tutor. So I'm answering two questions in this video, okay? And these questions, they are coming from a 2021 paper 2 for grade 12s. So I'll answer two questions in this video, and then in the next video I'll answer two more questions until we finish all the questions in this past paper, which is a 2021 paper 2 for grade 12s. All right, let us begin. Figure 1.1 and 1.2 shows freshly cut potato chips suspended in solution E and F at the beginning of an experiment and after 2 hours respectively. So we've got two figures here. We've got figure 1.1 and figure 1.2. So figure 1.1 is indicating how things were looking at the beginning, and then figure 1.2 is indicating how things were looking after 2 hours. So we've got solution E and solution A, and the potato chips that were suspended in these two solutions. All right, let's look at the first question. Explain the difference in the appearance of the potato chips in solution E and F after 2 hours. Okay, but vaisize sometimes ama conversation mushe. They are just asking you to explain the difference. The difference of the potato chips in solution F and E after 2 hours. So if you look at the potato chips that were suspended in solution E and F after 2 hours, you can see that they are different from how they were looking at the beginning, isn't it? So we need to explain the difference. How did this difference come about? Now, if you look at this question, there's osmosis everywhere. We can see osmosis everywhere here, okay? Osmosis is visible here. So the question is, what is osmosis, by the way? Osmosis is simply the movement of water molecules from a region of high water potential to a region of low water potential. Or you can simply say osmosis is the diffusion of water molecules. Okay. Now, another thing that you need to know is that a cell, whether a plant cell or an animal cell, it can be exposed to three kinds of solutions, which are hypotonic solution, isotonic solution, or hypertonic solution. Okay. Now, another information that you need to know is that hypotonic solution will cause a cell to increase in size, and then hypertonic solution will cause a cell to decrease in size. In short, it will cause a cell to shrink. And then isotonic solution does not have any effects on the cells, whether a plant cell or an animal cell. So let's now use that information as we are answering these questions. So question 1 is asking, explain the difference in the appearance of the potato chips in solution E and F after 2 hours. So if you look at the potato chip which was suspended in solution E, we can clearly see that it decreased in size. Now, what happened for it to decrease in size? It lost water, and it lost water by the process of what? Osmosis. Okay, that's what happened. It simply shrunk. Okay, it lost water. Kapela ni shrink chizungu. But what we can see here is that a potato chip which was suspended in solution E lost water. It shrunk. And if you are to do this, you can actually do this experiment. I'll show you how at the end. I'll tell you how you can do it. Otherwise, this is very much simple for you to set up. Otherwise, the potato chip which is in solution E shrunk, okay? It lost water. That's what's happening here. And then if you look at the potato chip in solution F, it actually gained. It gained water by the process of osmosis. It increased in size. We can see that there's an increment in the size. Looking at how it was looking at the beginning and how it's looking after 2 hours, we can conclude that it gained water by the process of osmosis. Simple. 4 marks niano hapa. So 4 marks. Simple 4 marks. 4 marks. Alright, question 2. Based on the appearance of the potato chips in solution E after 2 hours, what term could be used to describe solution E? What term could be used to describe solution E? Now, I said that a cell whether a plant cell or an animal cell, it can be exposed to 3 types of solutions, which is hypotonic solution, hypertonic solution, or isotonic solution. Now, hypotonic solution will cause a cell to do what? To increase in size. It will cause a cell to gain water, because hypotonic solution is a type of solution which has got high water potential than the cell. And then hypertonic solution, it will cause a cell to lose water or to shrink, because hypertonic solution is a type of solution which has got low water potential than the cell. Are you following? So now, isotonic solution, on the other hand, it has got no effect. It has got equal concentration with the cell. So the question is asking us to give a term, okay? They're asking us for a term. What term can we use to describe solution E? In short, what is solution E? Okay, that's what they're asking us. Well, solution E is simply hypertonic solution, because looking at what happened after 2 hours, these potato chips shrunk, meaning it was placed in a hypertonic solution. And like I said, when you put a cell in a hypertonic solution, what will happen? What will happen? What will happen? Excellent. It is going to shrink. And that's what happened. So solution E is hypertonic solution. That can be the term which can be used to describe solution E. It is simply a hypertonic solution. It has got low water potential compared to a cell. Okay. Next question, please. What would be the state of cells in the potato chips in solution in solution F after 2 hours? What would be the state of cells in the potato chips in solution F after 2 hours? So if you look at the potato chips in solution F, what happened? They gained, isn't it? It increased in size. Looking at the diagram after 2 hours, it increased in size. So they are asking us, tuma cells tomene tupanga I potato chips in solution F, tuone kabwanji, they are in what state? Okay, how are they looking? They are in what state? That's what they are asking us. Well, obviously they gained water. These plant cells, they gained water. Okay, these plant cells, a potato chips, these are plant cells. Okay, they are plants. So these plant cells, they gained water and they became fully badged. So they are in that state. They are fully badged. Okay, they gained water and became fully badged. If you put an animal cell in a hypotonic solution, which is solution F, by the way, solution F is hypotonic solution. So if you put an animal cell in a hypotonic solution, it will gain water and burst, because when you look at an animal cell, it does not have a cell wall, and the cell wall is what prevents a cell from bursting. So since an animal cell does not have a cell wall, it only has a cell membrane as the external barrier or the outermost layer, it is going to burst because the cell membrane is not strong enough to withstand the pressure. But if you look at a plant cell, it has got a cell wall which prevents that cell from bursting. So yeah, the cells, they're in a state of, they are fully badged because they gained water. So they gained water and became fully badged. All right, 1 mark. So they are fully badged. Let's look at the next question. State the importance of the state of cells in 3 above to plants. Oh, that's important for growth. They are fully badged, and that is important for growth. When plant cells gain water, okay, or when they absorb water, that will cause them to grow. Okay, that will cause if you check, the potato chip has increased in size, so it's important for growth. The state they are in is important for growth. Okay, and plants undergo 3 stages as they are growing, which is cell division, cell elongation, and cell differentiation. If you look at cell elongation, it is a process by which cells grow bigger by absorbing a lot of water. So when plant cells absorb water and become fully badged, that will result in growth. So it is important for growth. All right. Final question. Explain the meaning of the term isotonic solution. Well, an isotonic solution, this is a solution that has got equal concentration with the cell. Okay, it has got equal concentration. So if you put a plant cell or an animal cell in an isotonic solution, nothing will happen because the concentration is what? Equal. All right, let us look at question 2 for this paper. So table 2 shows various food tests carried out by Elena. So let's look at table 2. We can see that we've got nutrients there, we've got under nutrients we've got starch, and then we've got I had a teacher who used to do whenever there's a dash. So she would be like, Chanda, you understand? So under nutrients we've got starch, protein. Under test we've got emulsion, burette. Under observation we've got color change from brown to blue, black, white precipitate. Then under conclusion we've got starch is present, protein is present. Okay, so that's what we have under table 2. First question. Complete the table by filling in the spaces. So let's look at what is happening here. So, you know, when you're testing for starch, what is the name of the test as you're testing for starch? What is the name of the test? Well, the name of the test is iodine test. So where there's 1, you just put iodine, okay? Iodine, that's the answer, iodine, because the name of the test when you're testing for starch is iodine test. Iodine test, they actually call it the iodine test for starch. So pawano hapo nikufaka pochani iodine, okay? And then pali 2, what nutrient are they asking us? So let's look at the test. The name of the test is what? Emulsion. What nutrient do you test for when you're using the emulsion test? What nutrient are you looking for? So the answer probably 2, it is lipids or oils, okay? Lipids. When you're testing for lipids, you can use this test known as the emulsion test, okay? So the answer for 2 is lipids, okay? Lipids. And then when you under observation we've got white precipitate. So what does that mean? If we've got white precipitate, that simply means lipids are what? Lipids are present. Okay, that simply means lipids are present. So part 3, lipids are present. When you look at the observation, it will tell you the conclusion. So if you've got a white precipitate, that simply means lipids are what? Present. How did I know that it's lipids? Because of the name of the test. The emulsion test is one which is conducted when you're testing for lipids. So that's why I say, that's how I knew to say, oh, the nutrient they're testing for here is, the nutrients they're testing for here are lipids. Okay, and then conclusion, I knew that lipids are present because of the observation. Yeah, so don't get confused. Okay, nutrients, protein, then test burette, the burette test. What is the observation? We don't know, but we can tell the observation by looking at the conclusion. So they're saying that proteins are present, isn't it? Well, when proteins are present, then the observation would be a blue-black. There was a blue-black. Is that true? Is that true? No. I wanted to test you. When proteins are present, what are we going to see? What are we going to see? So when proteins are present, the solution turns purple, violet, okay? Purple or violet. Those are the colors that you're going to see, purple or violet. That's what we're going to see when proteins are present. So yeah, you can put purple, or you can say solution turns purple or violet, okay? Under 4, that's what you can put. So we've completed the table. 1, it is iodine test. 2, lipids. Then 3, lipids present, and then 4, solution turns purple. Okay, let's look at the next question. Which nutrient in the table would cause kwashiorkor if lacking in the human body? Simple. The nutrient is proteins. So proteins are important for growth. So when you lack proteins, protein deficiency can lead to kwashoka. Kwashoka is one of the diseases that can be caused when one lacks proteins in their body. Okay, simple. Name a nutrient not in the table which helps to prevent constipation. What nutrient helps to prevent constipation? Well, the nutrient is roughage. Roughage is the nutrient which helps to prevent constipation. Okay. And then the final question, state 2 deficiency diseases in plants. 2 deficiency diseases in plants. All right, so we've got chlorosis. Okay, chlorosis is a deficiency disease in plants. Umaona ngati tuma leaves tomonkala yellow vija, so that's chlorosis, and it is as a result of a plant lacking manganese. Okay, manganese is important. Okay, so when a plant lacks manganese, it will lead to chlorosis, which is a deficiency disease in plants. Another one is stunted growth. Stunted growth, that's another deficiency disease in plants, and this is as a result of a plant lacking nitrogen. Nitrogen is important for growth, so if a plant does not have nitrogen, it will lead to stunted growth. All right, so I'm done. I have answered the 2 questions from 2021. Okay, and this is the first video for 2021. Okay, so in the next video I'll answer question 3 and question 4 until we finish the whole paper. This has been Mr. Philip Kauma, the excellent biology tutor. I hope you've enjoyed this video. Stay blessed.